discussing lecture 10, which is culture media. Culture media is from basic microbiology. I got my finals tomorrow. Yeah, this is for like audio podcast only. Yeah. Quick disclaimer, this video is only for self-revision purposes. Examples used here are for like remembering techniques rather than actual examples. However, let me get started back. Okay, so since we're talking about culture medias, the criterias. Okay, first of all, what is a culture media? It is the food and environment where microbes, like which microbes need to like grow and survive. Okay. Yeah. The criterias that a culture media must meet. Like a culture media must meet. There are 6. First, we got right nutrients in the right amount for like specific. So you know how like every single human being have like different needs. For example, it's 33 degrees outside and I still need my hood. Why? Because that's just the way I was built. That's me, right? So microbes got that stuff as well. So the media must contain right nutrients for the specific microorganisms we want to grow at the right amount. Now, why is this crucial? If it's not in the right amount, we're probably not gonna get like best growth. And if it's more than the right amount, then there might be a negative impact, like cytotoxicity. Get it? So it's like coffee is good for you. I don't know. Okay, I don't know. But let's just say coffee. You have coffee. I have coffee a lot. Okay, I pretty much drink like 1 liter every single day. So let's just say you have more coffee than you really do. Like, I'm genuinely like a really sleepy person. So like when I have like I need coffee to survive. Okay, so like to make these videos, to yap and shit. But let's just say if I like up my dose, I might die because of like caffeine overdose, right? So yeah, that's a negative impact. It's like it's sort of like poisoning me, toxic, cytotoxic. Get it? So number 1, it must contain the right nutrients for the specific microorganisms we want to grow in the right amount. Number 2, it should contain sufficient moisture. Now, if we know one thing about microorganisms, is the fact it's moisture. Ugh. Okay, so bacteria usually don't survive in dry conditions, okay, or like dry surfaces. Then we got adjusted pH, right? So it should provide, the culture should provide adjusted pH. Now the pH has to be, I don't know how this makes sense, but it has to be really close. Like most of the bacteria, they have a pH which is like really close to the neutral pH, as in like pH 7. Get it? It is usually a little more than 7, but it cannot go below 6.8. Then we got the medium should provide suitable level of oxygen or no oxygen, depending on the microbe. Okay, so like a quick revision. What is exactly a medium? Medium is in a petri dish, like when they say we're culturing like an organism. So what you really mean is that you're farming microbes in a little petri dish, like those like little transparent bowl, thin bowl, Those are called petri dishes. So when you're farming microbes in there, that is called culture. Now, what's a culture media? So you can't just like get a random plastic container, like put it there and expect microbial growth. No, especially if you want like a specific kind of microbe to grow. What are you gonna do? You're gonna have to put food. It's like you bait them to come and just like be like, yo, I got your food here. You make a population there. It's like that. So that's media. Get it? You got right nutrients, right amount, sufficient moisture, adjusted pH, oxygen or no oxygen. So, like some microbes, they need oxygen, for example, E. coli, and some they don't. I will tell you examples of it later. So, depending on which microbe you want to grow, you put that kind of condition in there. Then we got the medium must initially be sterile, which means that there should be no living microbes present before, like imagine, okay, I have like this, I'm renting out a house to someone, right? So before I rent out my house to someone else, I gotta make sure that my house is empty, right? Because if it's not empty, how are the other people, like my new people who I'm like renting my house to, how are they gonna live? Right? I mean, it's gonna be like invasive of their like privacy, and they're not gonna be able to live as well, like, because of a lot of reasons. It's like that. So yeah, there should be no living microorganisms. Okay? It should be non-pathogenic, because let's just think, okay? In a petri dish, okay, where I'm trying to grow cats, but there's already pre-existing lions in there. I don't even know if lions eat cats, but if the lions are in there and they eat cats, the cats will not be able to grow because the lions are gonna eat them. Okay, and then lastly, we got growing culture should be incubated at the proper temperature at optimum growth time. If you don't give enough time for growth, how the hell are they gonna grow? Okay, then we got types of culture media. Now, there are 3 primary culture media, like 3 primary types of culture media. We got physical, we got chemical, we got functional. Okay, so physical, okay, this one really depends on the medium's normal consistency. Okay, so what is this? There are 4 types of physical state, like physical form of culture media. One is liquid, semi-solid, liquefiable solid, non-liquefiable solid. Get it? Yes, you do. Okay, then we got chemical culture media. It's got 2 types. One is synthetic chemical, which has a defined, like it is chemically defined, okay? And non-synthetic, it's not chemically defined. Now, one way to remember this is that, okay, when you make something in the lab, when you're generally making something, okay, you know what's in the process because you're making it. But let's just say something's given to you, like the same thing's given to you. You're not gonna know, like, what is. It's like, okay, you know how the chefs, they know what ingredients are used to like cook something. But like, I'm not a chef. I know what is used to like cook something. So to cook something is to synthesize. So the product is synthetic. So it is chemically defined because we know what's being put in there. And the non-synthetic, as in I'm not synthesizing the product. Like I'm just like going to the restaurant and eating. It is non-synthetic. And it's not chemically defined. Easy, right? It's easy. So these were like the two chemical types. Then we have functional types. Now we got general purpose, for like the public. Then we got enriched. Yeah, we're gonna read about it later. Then we got selective. Then we got differential. We got miscellaneous. Then we got specimen transport. 7 is assay, like assay, like ASSAY, no, essay. And then we got enumeration, which is pretty much just like count. So in total, in the functional thing, we got 8 of them. The core difference between, now we're going to talk about physical form of like culture media, okay? So the core difference between the liquid media and the solid media is agar. Those things that we see in like, those like really aesthetic desserts. Transparent. It's in your gelatin. Get over yourself. Okay? So let's talk about liquid media. Liquid media are defined as water-based solutions. So what really happens is it's made with dissolving various solutes in distilled water. Get it? Of course. Okay? Some other names for liquid media is broth, milk, or infusion. They do not solidify at room temperature because it's called liquid media. Like, get a life. Anywho, growth occurs throughout the container and can present a dispersed, cloudy, or particulate appearance. So we don't really like count in this type of media. Why is it? It's because you can't. You only get to see like, it's a mosquito. Okay. You only get to see like if there is microbial growth actually happening. And the reason why we still do like liquid, we still use like liquid culture is because to see if like the thing, like the microbial growth, it's gonna work in the solid one or not, like the solid agar or not, to observe if like there's growth occurring for the starch culture, to see whether or not there is there are microbes present. It's like let's just say I wash my hair on very rare occasions, but like I wash my hair, right? And I'm just there like, I'm the cleanest person ever, like shut up, right? But one of my friends are like, no you're not. And then she like swabs my hairline, and then she puts it like in a liquid broth, the drill. And then you get, if there's like cloudy stuff, that means there's microbes present, like depending on like the culture you've prepared, like she's prepared. So it's like that. Subculture. Now let's talk about subculture because this is kind of important. Subculture is like, let's just say okay, it's like you already have like grown a bacterial population in like a certain culture, okay, and then like you feel like it's getting overcrowded, or like you just want to look into it like more closely, so you just like get that sample and like a fresh sample, so like you can accurately observe more of it, okay, in like less population. That like the whole transporting from like an old culture to like a new culture, like a fresh culture, is called a subculture. Another thing is for first inoculation. What is inoculation? It is adding microbe into media, like whether or not there is microbe present. That's it. That's why you want to use it. Number 4, when you don't know the bacteria will survive in a solid agar. We talked about this. Number 5, when there's no need for enumeration. Now, what do liquid media really need? They need an aeration system. Okay? It sounds very fancy. It's not. What is an aeration system? So since it's liquid, okay, and it's like, you know, you're gonna mix like a couple of shit with like the liquid, so it's gonna be quite dense. You know, you're not sure if oxygen is gonna like get inside, especially for, you know, my aerobic bacteria or like aerobic microbes. If oxygen doesn't get inside the liquid media, it's gonna be a really bad growth situation, right? So that's why the incubator, I don't know what an incubator is, I'm guessing it's like a test tube, it's shaken so oxygen is provided in the media, the liquid media. This is a mechanical way to increase the O2 level in the liquid media, and this is not done in solid. Why? Because, you know, in solid media, like it's pretty much just like in contact with the oxygen. It really doesn't have to like go deeper and like dissolve. Like, you know, liquid, you're not sure if the oxygen is going to dissolve or not. That's why you just gotta shake it, which we call the aeration system. It's not used in the solid media. Now, for the semi-solid media, now this is quite interesting considering it's not really liquid, nor is it a solid. Now, for the semi-solid media, you want your agar concentration to be somewhere around 0.3 to 0.5%. It is done, like the whole thing is done to check the motility of microbes, whether or not it can move or not. So how do these microbes move in this type of culture? They swarm or swing. Swarming is a crawl-like movement, and it is done when the tail is usually inactivated. When during ordinary room temperature, semi-solid media exhibit a clot-like consistency because they contain an amount of solidifying agent, which is usually agar or gelatin in 0.3 to 0.5%, which thickens them but does not create like a firm substance. Get it? So these this motility test that we just talked about, it cannot be done in the solid media. Now there are 2 types of solid media. Before we talk about the 2 types of solid media, let's talk about solid media in general. It provides a firm surface on which cells form discrete colonies and are advantageous for isolation and subculturing bacteria and fungi. It's also really good for enumeration, which I'm guessing is like counting the micro. And let's talk more about this because oh my god, it's like 12 a.m. I'm gonna sleep. I'm on finals tomorrow. So okay, so like the agar here is like 1 to 5%. Okay, 1 is like realistically used. Okay, no one really uses like more than 1 because I don't know. 5 is the highest they would go. So solidifying media, it's called 2 types. One is liquidifiable, one is non-liquidifiable. Okay, it's like, how do I explain this? Okay, so imagine that there is solid. This is a solid media. This is a solid thing. This is a solid thing, right? If I melt this, okay, can I just like get the same product if I re-solidify it? No, right? Because it's got 2 kinds of materials and like a bunch of screws and shit. We're not gonna get it. It's not gonna be the same. But for this, okay, let's just assume the screws here don't exist. It's gonna be the same. Okay, so liquefiable is the kind of this. It's like this. Okay, so you make a media in like bulk amounts, which give the same results as fresh. No difference in quality. So okay, I feel like you can just like see I'm reading from like this. I don't like that, and I also like not very classes. So let's talk. Okay, so it's for bulk. So imagine like you gotta make culture media every single day. It's like meal prepping, right? So you gotta make a culture media every single day, and it's the same media all the time. So you make it in bulk. You use it you make it in bulk. You take a little bit of it, you liquefy it, or like you solidify it. You use it, all the crap. Right, like you can like freeze it and use it and freeze it and use it until you're done. It's like you got like a big thing, you take a little bit of it, use it, and then you throw it off. Use it, throw it off. It's like liquid. Okay, but 52 degrees Celsius is the temperature at which liquefiable solid media like agar is cooled down before pouring into petri dish. Okay, I have literally no idea, but I'm just gonna associate 52 degrees Celsius with like liquefiable solid media. I don't know what the rest means. Anyway, for the non-liquefiable solid media, what do we do? Usually, examples of this is potato slice and meat extract. I don't know how it's gonna relate to any of that, but let's talk about it. It's like you cannot use it. Like you cannot just like freeze it and use it. It's like, so you know how ice cream. Okay, yeah. Liquefiable solid media can be talked about like ice cream. Okay, so with ice cream, you take a little bit of it, you have it, and then you can put it in the freezer. You can have it whenever you want. But let's just think of the same thing as rice. Okay. After like a few days, you're not gonna have that rice, right? Because it's gonna, I don't know, ferment. It's kind of gonna get bad, right? I mean, even though it's in the refrigerator, it's like that. So non-liquefiable solid media has to be used within 3 days because it usually deals with antibiotics. And antibiotics are temperature sensitive. Now, why are antibiotics used? Antibiotics are used to prevent pathogen in media. Like I said, you're growing one type of bacteria. Now, this type of bacteria, it could get infected from other kinds of microbes. Very normal. So, in order to prevent that, antibiotics are used. And if antibiotics are used, you gotta use it within 3 days. Now, the presence. Why does the presence of pathogen matter? Let's talk that in formal language. It is because the presence of it causes media to lose efficacy due to temperature change. Efficacy means pretty much, I'm guessing, accuracy, efficiency, and when you're using liquefiable culture media, solid culture media, you gotta do the plating immediately, because you cannot just the moment you put it in, it's gonna solidify, right? So so the moment you put it, let's just say you're making gelatin, right? So you want to make gelatin in those really pretty, those cup, those muffin things, right? But imagine if your gelatin just solidifies in your non-existing. It solidifies in your non-aesthetic pants. How are you gonna feel about it? Exactly. That's what you're gonna do. Immediate plating. So that was non-liquefiable and liquefiable solid media. Now let's talk about. Oh wait, we didn't talk about liquefiable solid media all the way through. Hold on. Let's talk about liquefiable solid media. The one we can ice cream. The one I compared with ice cream. Now this one is a reversible solid media with solidifying agent like agar, which is a complex polysaccharide isolated from red algae gelidium. It's so the liquefiable solid media, its physical properties change in response to the temperature. Then we got any medium containing 1% to 5% and usually 1.5% of agar usually has the word agar in its name, and up to 45 degrees Celsius of the whole, when it's 45 degrees Celsius, it's semi-solid, and if it's less than 42 degrees Celsius, it's solid agar. No, okay, this is self-revision. When media is made in bulk, this media is made in bulk. Oh wow. This is for liquid solid media. 52 degrees Celsius is the temperature at which liquefiable solid media like agar is cooled down before pouring it into Petri dish. 52 degrees Celsius is the cooling down temperature. Then we got benefits of agar are numerous. It is a solid at room temperature and it melts, liquefies at boiling temperature of 100 degrees Celsius. Okay, now that makes sense. Once liquefied, agar does not re-solidify until cooled to 42 degrees Celsius. So it can be inoculated and poured in liquid form at temperature 45 to 50 degrees Celsius that will not harm the microbes or the handler. So let's give you some wisdom. First of all, please never do this. I don't know what you were thinking. Anyway, first the agar, okay, is solid at room temperature. Okay, it's melted at 100 degrees Celsius. Then it does not re-solidify on its own. Okay, you gotta cool it down to 42 degrees Celsius so it can be poured in the liquid form at 45 to 50 degrees Celsius, so it will not harm the microbes or the person who's handling all of this. So, for example, nutrient agar. So, we got non-liquefiable solid media, the one I compared to with rice. These are usually potato slices, meat extracts. I don't know why did I write that, but anyway. These materials include such things as rice grains used to grow fungi. I'm guessing it's an example. Cooked meat media, good for anaerobes, and potato slices. All of these media will start out solid and remain solid after heat sterilization. Why did that remind me of Reddy's experiment? Like Francis Reddy, who proved that the abiogenesis thing wrong. He did do that. I do not like my side. Let's talk about chemical composition of media. So the one where the chemicals are like defined, those are called synthetic. Such media contain pure organic and inorganic compounds that vary little from one source to another and have molecular content specified by means of an exact formula. Complex or non-synthetic media contain at least one ingredient that is not chemically definable, not a simple pure compound, and not presentable by an exact chemical formula. For example, infusion media. A very easy example of this, I would say, is that how we can pretty much recreate a lot of the non-living things in lab because we know like what amount of what is present. For example, methane gas, right? We know how much carbon is used, how much hydrogen is used, so we can recreate that in a lab, right? So it's synthetic. However, things like living things, okay, like human beings, okay, we know like livers and heart, everything exists, but there's one thing that is still not figured out, is the little line between the very little line between a living and a non-living organism. We know it's not DNA because DNA is technically not a living thing, but it's that thing that makes us a living thing. So you see, because of this one thing that we don't know, we cannot synthesize it, right? It's like that. So for non-synthetic media or complex, at least one ingredient has to be non-chemically defined. So like it cannot be represented in exact chemical formula. For example, infusion media. Then we got functional types of media. Okay, so there were 3. Let me do a quick recap again. So we got the initial media types, like the primary, 3 primary media types were physical, chemical, functional, right? From physical, we got 4 types of media, which is liquid, semi-solid, solid liquefiable, solid non-liquefiable. For chemical, we got 2 types, synthetic, non-synthetic, right? For functional, we had around 7-ish things, we got 8, okay? General, enriched, selective, differential, miscellaneous, specimen, transport, assay, and enumeration. So let's talk about general purpose media. So every type of microbe can be grown here. So let's just say you're like a noob at this microbio thing. like me in the lab. So you just want to see something. Doesn't matter what kind of microbe you want to see. You just want to go and you want to see something, like some kind of like an action, right? Like you want to see like whether or not you can actually like make microbes, that thing. So it's like that. We're probably gonna like do the whole general purpose media instead of like a very specific thing. Every kind of microbe can grow in there. Like it's got a very broad spectrum of microbes. It is usually, as a rule, they are non-synthetic, contains a mixture of nutrients that could support the growth of pathogens and non-pathogens alike. Examples being nutrient agar and broth, brain heart infusion, trypticase soy agar. TSA. Trypticase soy agar is a type of general purpose media which falls under the category of functional types of media. This is used to count the total number of microbes present. Then we got enriched medium. Enriched medium contains a complex organic substance such as blood, serum, hemoglobin, or special growth factors such as specific vitamins, amino acids that certain species must have in order to grow. Bacteria that require growth factors and complex nutrients are termed fastidious. Okay, so if you're a picky eater, you're fastidious. My glasses, I'm sorry. Fastidious. If you're a picky eater, you're most likely fastidious. Blood agar, which is made by adding sterile sheep, horse, or rabbit blood to a sterile agar base, is widely employed to grow fastidious streptococci and other pathogens. So in enriched medium, we understood that like a specific nutrient is enhanced. It's like playing baiting, So you want to bait me? You put waffles and desserts in your house, and you just go, "Hey, I got candy," and it comes. It's like that. Enriched medium. Okay. One way to remember this is I would say, this is good. Blood agar. Let's talk about blood agar again. It's made by adding sterile sheep, horse, or rabbit blood to sterile base. Is widely employed. Okay. Then we got selective media, which is also another kind of functional type media, culture media. So it's got one or more agents that inhibit the growth of certain microbes or microbe, but not others, and thereby encourage or select one microbe and allow it to grow. Now, with enriched media, we were like baiting people into like doing stuff. With selective media, what you do is you cut it, okay? So it's like a job interview. You don't give the job, you don't get the job. Yeah, it's like raising standards. If you fall into that standard, you're probably gonna it selects. Get it? It selects. So instead of just like, okay, so imagine the microbe wants to grow. Instead of just like providing what it needs, what it does is that it does a preventive. Like it creates an environment where the microbes will. It's like instead of your friend saying that, dude, okay, so you should do this and that to just like become a better person. Your friend creates an environment for you where you will do all of this and you will like give up bad habits on your own. I don't know if it makes sense, but it makes sense. Shut up. So in non-selective, it's like very much the opposite of like the general purpose one. So in general purpose, every single one of the microbes can grow, but in selective ones, only the selective microbes can grow. So here EMB is used for E. coli. So figure, it shows heavy mixed growth of gram-negative and gram-positive bacteria. This one. Bacteria including Enterococcus species left. This is left. Okay. Oh no. This is. Oh no. Yeah. Okay. The selective media, for example, phenyl ethyl alcohol agar, only allows the Enterococcus species to grow. Right. So you see, this is mixed. This is selective. So selective media are very important in primary isolation of specific type of microorganisms from sample containing dozens of different species. For example, poop, saliva, skin, water, and soil. So mannitol, salt, agar, media contains concentration of NaCl, which is 7.5%. That is quite inhibitory to most human pathogens. Most human pathogens cannot grow in 7.5% of NaCl concentration, which is inside mannitol, salt, agar, MSA. Okay? Oh no. Okay. One exception is the genus Staphylococcus, which grows well in this medium and consequently can be amplified in very mixed samples. Staphylococcus grows well in this medium and amplified. Okay, this is the amplified. The reason we use selective media is so we can study one microbial growth in isolation. Then we got differential media. Differential media employs some factors that allow colonies of one bacterial species to exhibit certain metabolic or culture characteristics that can be used to distinguish them from other bacteria growing on the same agar plate. Okay, so it's like it shows visible differences between different types of bacteria, like color changes and color appearances. Shows differences based on bacterial metabolism. It's like in one culture you can make you can grow two types of microorganisms, but the differential media, it will like differentiate based on the bacterial metabolism. Example of this, McCockney agar, which differentiates between gram-negative bacteria and that can and cannot ferment sugar lactose. So when 2 gram-negative bacteria grow, the lactose ferment bacteria are usually pink and non-ferment bacteria are colorless, slightly pale. So what does this differential media contain? Okay, it contains indicators like pH indicators that can change color based on bacterial activity. Okay, no, this is not number 5, but okay, so there is another kind of bacteria. So remember selective media. Selective media, we talked about how we eliminate the other pathogenic microbes by like making the conditions very much like harsh to them and not the one that you want to grow. So that combined with differential medium, which we just like talked about like right now, there is another media called selective plus differential media. So sometimes selective and differential characteristics are combined in a single medium. Okay, suppose we want to isolate the common bacterium Staphylococcus aureus found in the nasal pathways. The organism has tolerance for a high concentration of sodium chloride, so it can also ferment the carbohydrate mannitol to form acid. So differential media is used. Mannitol salt contains 7.5% of sodium chloride, which will discourage the growth of competing organisms and thus select for the favor of growth of S. aureus. The salty medium also contains pH indicator that changes color if the mannitol in the medium is fermented to acid. The mannitol fermenting colonies of S. aureus are either differentiated from colonies of bacteria that do not ferment mannitol. Right? And we got miscellaneous media. Now, miscellaneous media is of two types. One is reducing media, one is carbohydrate fermentation media. Now, what is miscellaneous media? It's weird, special purpose, or oddly specific media, which occurs for like reducing. Reducing media. Let's talk about reducing media. So, it contains a substance such as thioglycolic acid or cysteine that absorbs oxygen or slows the penetration of oxygen in medium, thus reducing its availability. Reducing media is important for growing anaerobic bacteria or determining oxygen requirements. Another type of miscellaneous media is carbohydrate fermentation media, which contains sugar that can be fermented to acids and a pH indicator to show these reactions. Many other media are used for other biochemical reactions that provide basis for identifying bacteria and fungi. So reducing media is usually used for anaerobic bacteria, and carbohydrate ferment ones are used for identifying bacteria, which are biochemically different, I guess. And then we got transport media. So in transport media, are used to preserve specimens that have to be held for a period of time before clinical analysis or to sustain delicate species that die rapidly if not held under stable conditions. So remember COVID-19 tests that we did? Okay, so they would take our cotton swabs and then they would just take it to the hospital. So from the home to the hospital, the material could be destroyed. So it's a culture is prepared, which is for the transport only. Here the buffer used is called PBS, makes the bacteria constant. Examples of transport media is Stewart and Ames transport media, which contains salt buffers and absorbents to prevent cell destructions by enzymes, pH, and toxic substances that will not support growth. Then we got assay media. Assay medias are used by technologists to test effectiveness of microbial drug and by drug manufacturer to assess the effect of disinfectants, antiseptics, and cosmetics and preservatives of the growth of microorganisms. So I'm guessing it says imagine you're a pharmacist and you have developed this new product and you want to see if it kills microbes, right? So how are you gonna just see if it destroys microbes without destroying or reacting with the culture media? You use an assay media, I think. Then the last one, oh my god, I'm so happy. Oh, I have been yapping for 2 hours straight and I also sang for God knows how many times. Okay, anyway. Then we got enumeration media, which is used by industrial and environmental biologists to count the numbers of organisms in milk, water, food, soil, and other samples. I'm not even going to explain that. Okay, let's talk about PBS, which is related to transport media, like the transport urea. Media uses PBS as a buffer for constant bacterial thing. So PBS is actually phosphate buffered saline, which is buffered salt solution maintaining pH and osmotic balance that doesn't kill or grow bacteria, just keep them stable. That's it. That was lecture 11. I would have covered that was lecture 10. I would have covered lecture 9 too, but I think we're just gonna have to read all that. Anyway, best of luck, sweetie. You got this.